Raquel Portela is a molecular microbiologist researcher at IMIB Lab. Currently, Raquel studies the key mechanism underlying the host-bacterial infection interplay, using Staphylococcus aureus as a model organism and the effects of antibiotics on this interaction. Focus on the behavior of Staphylococcus aureus in the context of infection, its recognition by the host immune system, and the triggering and unveiling mechanisms of bacterial persistence. For this, she employs advanced imaging techniques such as super-resolution structure elimination microscopy. Thanks once again to have accepted our invitation, and you can start.
Thank you. You can hear me, right?
Yeah,
okay.
Yeah.
Now I'm in full mode
Yes.
screen. I just need to take this from here, otherwise I can see. Okay, so thank you very much. And first of all, thank you for having invited me to present the work that we will start implementing here at ITQB under the scope of this MPS NOVA research project. So it will be a bit different than the work that Madalena showed with a lot of very nice presentation with a lot of results. We are now starting to implement the technology. So I will talk about modeling bacterial pneumonia, infection dynamics in lung on sheep, a comparative study of Legionella pneumophila and Staphylococcus aureus. Okay, so first let me start by introducing to you pneumonia that all of you know, that is an acute respiratory infection of the lungs. Namely, it affects the alveoli and the distal airways, the bronchioli. And pneumonia and bronchiolitis, which are lower respiratory tract infections, in 2019 were responsible for almost 500 million people affected globally, with a mortality of more than almost 2.5 million deaths, which turns pneumonia with a high mortality disease, and is the largest infectious cause of death in children. So it is considered a mild to life-threatening illness and can be caused both by viruses or by bacteria. And of course, the risk factors are, like in most of the diseases, the age of the patient and the pre-existence of other comorbidities. And to worsen the scenario, it can be spread by direct contact with other infected people. So epidemiologically, pneumonia can be divided in two different types according to the settings in which the pathogens were acquired. If it was in the community, it's considered community-acquired pneumonia, and if it was in the hospital-acquired pneumonia. And inside the group of hospital-acquired pneumonia, then the distinction is made based on the fact of the patient have been interned into the intensive care units or not, and if it was being ventilated or not. And this division in community and hospital acquired is not only because of the settings in which the disease was acquired, but mostly because usually it refers to different pathogens and different progressions of the disease that this represents. So if we talk only about pneumonia caused by bacteria, which is the ones in which we are interested. In hospital-associated pneumonia, what happens is that the majority of these infections are caused by Staphylococcus aureus. And this is related with the fact that Staphylococcus aureus is a highly resistant pathogen that thrives in the hospital environment. And so with the comorbidities associated to these patients, it's easier to acquire these bacteria and to develop pneumonia with this bug. If we talk about community-associated pneumonia, what happens is that the major agent causing pneumonia is Streptococcus pneumonia, which supplements by far the numbers of the other pathogens, being followed by pneumonia caused by Legionella pneumophila, Staphylococcus aureus, and Mycoplasma pneumonia. That follows the Streptococcus pneumonia. And we are specifically interested in studying these two pathogens, Legionella and Staphylococcus aureus, because among the intracellular pathogens that cause the disease, and we are specifically interested in follow the intracellular pathway of these pathogens, are the two more different, as I will show you, that causes the most severe forms of the disease. So what is interesting and important is that different pathogens cause different clinical manifestations of the disease. And they are very different. So Staphylococcus aureus is this gram-positive cocci bacteria, highly resistant to antibiotics, and that causes a typical pneumonia that is characterized by a well-defined lobar consolidation here in just one lobe of the lung. And what is affected is usually the alveoli, and it can cause a disease, a necrotizing pneumonia, which involves the necrosis of the lung tissue. On the other hand, Legionella pneumophila is a gram-negative and rod-shaped bacteria that causes Legionnaire's disease. You probably all heard about these outbreaks, and it causes an atypical pneumonia in which the tissue that is affected in the lung usually is the interstitial tissue surrounding the alveoli. And in the case of Legionnaire's disease, it is usually linked to other extrapulmonary manifestations, not only in the lungs. Okay, so we study bacteria, and why are we interested in study, and what are we interested in study from the bacterial perspective in this context? So we are interested, and I work with Staphylococcus aureus for a long time ago, and I'm interested in studying the intracellular life cycle, both of Staphylococcus aureus and of Legionella pneumophila in the context of this disease. And we have been dedicated a long time to study the roadmap of infection of Staphylococcus aureus. And what is known is that Staphylococcus aureus invades different types of cells, epithelial, endothelial, immune cells. And when they enter the cells, they reside inside compartments called endosomes or phagosomes that then get matured, they maturate, and they fused with lysosomes in order to be targeted, the bacteria to be targeted to destruction. And Staph aureus has two main strategies to evade this death by the host cell. In phagocytic cells, typically, Staphylococcus aureus has the strategy to withstand the conditions inside the cells. They replicate inside this vacuole until they have the trigger to destroy the cell and to promote secondary infections upon their release. In non-phagocytic cells, typically what happens is that they can escape and evade from these compartments to the cytosol of the mammalian cell, and then they replicate there until they promote the same cell death and the bacterial release. But there are a lot of underlying mechanisms in both of these pathways that are not known and that we are interested in understanding, and namely in the context of pneumonia. In the case of Legionella pneumophila, what happens is that Legionella enters into the cells, most often to alveolar macrophages, although they can also invade epithelial cells. And they reside also in these vacuolar compartments, Legionella-containing vacuoles, where they inhibit the fusion with lysosomes and they recruit other proteins, and they can hijack mechanisms of the host cell in order to take an advantage to proliferate and to cause disease. Another feature, intracellular feature in which we are interested is in the formation of persisters, which is common to both of these pathogens. And in persistence, what happens? So persisters are a small fraction of the population of bacteria that enters in a state of dormancy, of a metabolic inactive state, in which what happens is that the majority of the population is killed in the presence of an antibiotic, but then there are this fraction of cells that is not killed by the antibiotic, and so they continue inside the cells until they have the trigger to resume growth and promote again disease. And what is the clinical problem of persisters? The problem is that they contribute to the chronicity of infection, since we cannot eliminate all of the bacterial cells causing infection, and they can disseminate within the host, like in the case of macrophages that travel along the body to different sites of infection. And in these conditions, they can also avoid the antibiotic challenges. And so we are interested in establish the underlying mechanisms of persistence for both. But of course, that although we are interested and we study the bacteria, we are interested also in study this disease in terms of the host perspective, and why and how, and oh, sorry, this is, and the question is that we know that the host triggers alterations in the bacteria, but we also know that the presence of the bacteria will modulate the response of the host. And so we can see, and this is our punchline for this project, that different clinical manifestations, and we see that the different progressions of the disease result from the pathogen-specific interactions with the lung environment. And it is here that we need to upgrade our study to study also the host, to mimic this environment. And why? Because as all of you know, and Madalena resumed so well, we need to mimic the physiological conditions that we have in the lungs. So we have here the alveolus that are surrounded by these capillars, in which we have different types of cells that are interconnected, and in which the response to a pathogen involves all of this communication between the cells and not only one. So what we think is that if we find the common features that underlie the infection by both pathogens, we will reveal the mechanisms related to the infection severity. On the other hand, different features, so the ones that are specific for each pathogen, will reveal the drivers of the distant clinical manifestations that we have. And that's why we want to study the infection dynamics of these two pathogens in physiological conditions. But at what stage are we now? So we haven't started the implementation of microphysiological systems in our study. And before, we need to validate all of our studies in 2D cultures. And we have been dedicated in my work and in our lab and to the study of the division of Staphylococcus aureus. And why? Because it is very well known. It was described here at ITQB by the group of Mariana Pinho, how Staphylococcus aureus divides in planktonic conditions. And what they did is that they stained the membranes of the bacteria with Nile red and the DNA with Hoxt. And then they classified the phases of the progression of the cell cycle of Staph aureus according to the stage of formation of the septum. And then they quantified the number of bacteria that appears in each phase. And this works as a proxy for the time spent by the bacteria in each phase. So what we want to know is if the cell cycle progression intracellularly is similar to what is already described in planktonic conditions. And so what we have been doing is to create a system that mimics this system that was already performed. But since we cannot stain the membranes of bacteria, since we have an additional layer of membranes of the mammalian cell, we do this using a mutant strain of Staphylococcus aureus, in which we have a transmembrane domain of a protein fused to a fluorescent protein, M neon green, and the DNA binding protein, HU, fused to a different fluorescent protein, M scarlet. Then we infect our cultures and use super resolution microscopy to be able to detect the same stages of formation of the septum. And with this, we are able to evaluate the cell cycle progression for different cell types and with subcellular and time resolution. And just to show you a bit what we have been doing so far. So we started this by analyzing if the load of bacteria interferes with the cell cycle progression of Staphylococcus aureus. And so far we found that we see no difference in the cell cycle progression has a response to the different numbers of bacteria infecting the cells, although we know that it is described that the loads of bacteria interfere with the progression of the disease. But I also told you that we are interested in study persistence, and in this scope, what we do is to evaluate the same progression of the cell cycle, but in persistent conditions. And what we do to trigger persistence is to challenge the bacteria intracellularly with high concentrations of antibiotics. In this case, is 50 times the minimum inhibitory concentration of an antibiotic known to induce persisters. And so what we see here is the fraction of the population that resisted for 24 hours to this antibiotic, and so they are persister cells. And in this case, if we evaluate the progression of the cell cycle, we see that we have a delay in the splitting of these cells. And another example of what we can see is that using super resolution microscopy, we see that in the case of these persister cells, we found alterations in the phenotype. We see these D-shaped cells that we don't see in the absence of the antibiotic or for the beginning times post-infection. And what we see is that persistence includes an alteration of shape of the bacteria, which indicates that since it is the structure that is recognized by the host, this transition for persistence will also be linked to an alteration in recognition and in the modulation of the host that we want to further study. Regarding the study of Legionella pneumophila, it has been a bit different and mostly dedicated to the bacteria in terms of its manipulation of the host, which is probably linked to the fact that since the beginning, Legionella pneumophila was considered as an intracellular pathogen, which did not happen to Staphylococcus aureus. So, each Legionella bacteria is known to contain more or less more than 300 effectors. And the effector proteins are proteins that are translocated from the bacterial cytosol to the host cell cytosol using a type IV secretion system. And many effectors were already described, and it is known the mechanisms that with which they interfere in the mammalian cells, namely in macrophages, to hijack these processes to get an advantage to the cell. So in fact, what we want to do is to translate the findings from one pathogen to the other. So in Staphylococcus aureus, the majority of the studies concern cell division and the intracellular cell division that we are now trying to address. On the other hand, in Legionella, and we know much less about the manipulation of the host, while in Legionella pneumophila, it happens the opposite. So a lot of things and processes are known about the manipulation of the host cells, but almost nothing is known about the cell division of Legionella pneumophila. And so here, what we want to do is precisely to translocate our knowledge in order to use what we know from the point of view of cell division and to study cell division to apply to Legionella pneumophila and to understand what is done in Legionella pneumophila regarding the manipulation of the host in order to study the manipulation performed by Staph aureus. And of course, keeping this interest in persistence that we will keep for both pathogens. So what's next? So the point here is that that I show you was what we are proceeding in our lab. And next, what we want to do is exactly to apply the study of infection dynamics of these two pathogens using lung-on-chip to replicate, to mimic the condition in the lung. And for this, I was fortunate enough to win a staff exchange grant from the MPS NOVA project, which I acknowledge, and I will be visiting the lab of Professor Alexander Mosick since the middle of June to learn how to manipulate and to implement this system to then implement here at ITQB. And this system will be crucial for us because, as you see, we will have the hypothesis to have both epithelial cells, endothelial, and the macrophages in the same environment in order to study the infection dynamics of both pathogens. And moreover, this allows the air-liquid interface will be crucial to mimic the environment in the lung that we need to study. So with this, what we aim to do is to study the impact that both pathogens have in immune activation, in the cell viability of the host and of the bacteria, how they are cleared too. And the point is also that this system, this technology is compatible with multiple readouts, including advanced microscopy, which is very good for us since we already have these facilities to couple with this lung-on-chip technology. And this will allow us to study also the tissue morphology and to then manipulate this interplay host pathogen with different antibiotics to understand how this is altered in the presence of these antibiotics and in persistence. And we hope that in future we can have a platform for the study of infections and for probably to implement the study of new antimicrobial therapeutics necessary for counteracting pneumonia. So finally, I just would like to acknowledge to the laboratory where I am of Pedro Matos Pereira, the IMIB lab, and to Pedro Matos, which is here. Also to our collaborators, Dr. Alexander Mosick, which is here, and that I will visit shortly, and Irina Franco, which is our collaborator and expert in Legionella pneumophila. And of course, I would like to acknowledge all the funding that will allow us to implement this technology here, all the support of our institutions and the funding. And thank you very much. I'll be happy to answer to your questions too. Thank you.
Thank you so much, Raquel. Are there any questions? Yes.
Thank you very much, Raquel. To this very nice presentation. I have a question of the infection with Staph aureus and Legionella. For the different cell types that we have, for example, in the lung, so anaphysis, epiphysis, and the macrophages, to what extent do you think this is an active process that has been triggered by the pathogen itself? And what is the contribution of a, so to say, passive effect where the host cells are taking them up and the pathogen is remaining? Because in our hands, we see that as soon as we integrate macrophages in our systems, virtually 99% and more end up in macrophages. They are remaining, persisting, and from there they're spreading. And what we do not know is this something where the pathogen is targeting actively these immune cells, which are known for Staph aureus at least being representing a niche for persistence? Or is this something where Staph aureus is simply taken up by phagocytosis, and it would be take up any bacteria, and there it adapts and then starts to spread? And have you any experience on how this differs among, for example, clinical isolates that use different virulence factors?
Okay, well, what I would expect, in fact, is not easy. I don't know exactly what to expect, but what we know is that regarding your first question, like yes, both pathogens have the ability to infect different types of cells. And yes, I am expecting that we would like to do not in co-infection, but first start with seeing what is the the choose of each pathogen, where the tropism is, to where they go. And I don't know if it is a passive mechanism, because even in the same cells, sometimes they choose to do different things. Like in the case of Staphylococcus aureus, they can invade both macrophages or epithelial cells. Sometimes they can escape the vacuole. Sometimes they proliferate inside the cytosol, and this can happen in different types of cells. What we know so far is in monocultures. I don't know how it will be the difference if we have all the system involved. That I don't know. I would expect to have some tropism of the Staphylococcus aureus for maybe Legionella for macrophages. Probably, yes, that's what we have been seeing. But I don't know if Staph aureus will not spread all over the place. Concerning the clinical strains, what is known for Staphylococcus aureus is that it seems that all of the clinical strains that were tested, and there is a huge study about that, all of them can infect all types of cells that were tested, different types of cells. So I think it's not like just being undertaken and then they passively are free. You know, I think that they will modulate the host. They will hijack something to choose what to do. I don't know exactly what I hope to see. In terms of persistence, I don't know if I get very well what you were trying to ask me, sorry, but in persistent, may you repeat, please?
I mean, for the persistence, we see that in macrophages for at least 48 hours they can persist, and there is this bottleneck theory that has been brought up by Paul Cubes and others, which basically describe that there is an active phenotype enrichment, which, so to say, renders them to the most fittest phenotype, which is depending on the host cells where they are persisting therein, and then they acquire the ability to say disseminate among other immune cells. And how long do you see is there any data from humans in vivo how long they are persisting in a host cell, staying quiet before they're starting to disseminate?
I don't know how long they stay until they start disseminating, but I have read papers showing that they can persist for a long time inside macrophages and traveling along the body. Inclusive, they can go to other places like the liver, where they should be taken by a cup for cells, exactly, and so on and so forth. In fact, from the literature, I think they can withstand for a long time. But my experience is not that, just because I started doing infections in macrophages, and more or less about 6 hours I have no macrophages, they died. But I think that I started my experiments with loads of bacteria that may not be the ones that should recapitulate what happens in vivo. So from my data, I cannot tell that because I killed all the macrophages. But from the literature, we know that in other conditions, we should have the possibility to have persisters in macrophages for several hours or even days, I think. So probably, I think we can challenge them to be persisters and then see what we have in terms of dissemination. I don't know.
Thank you.
Thank you. Are there any more questions? I don't see it. So thanks once again, Raquel. Thanks all the participants that have stayed with us and in our next MPS virtual seminar.
Thank you very much.
Thank you.
Bye. Bye-bye.
Bye.
Bye.